Everyone hear me all right? So today I'll be talking about how Drosophila furrow invaginations are controlled by both cortex and membrane dynamics. And so on the left-hand side, I'm showing what is an early stage embryo of the fruit fly, and it's a single cell at this point. And over the course of about 60 minutes, you have 6,000 cells form an epithelial sheet around the outside of this embryo, effectively going from single cell to tissue over the course of 60 minutes. This eventually becomes a fruit fly. And so what I'm going to show here is a brief video that kind of gives an understanding or a feel for this. And initially you'll see on the top and bottom, you'll see these white lines coming in. Those are the furrow invaginations that eventually will become individual cells. And so they begin somewhat slow, and then they'll pick up pace and they'll start to go a bit faster. That is the behavior we are looking to model here. So in the top of this cartoon, circled or boxed in this green line here, we have just a cartoon of this, and we have a furrow or an embryo on the left-hand side where we have a furrow invaginating, and we're kind of zooming in. And on the right-hand side, we see the complete invagination. So if we look below that, we have a cartoon of this. And once again, this is a partial forming epithelial cell. We kind of cut it in half just for symmetry in this case. And we're going to use the velocity of this furrow invaginating to understand some of the underlying cellular mechanisms that are happening. And so I have a cartoon image shown to the left of the screen. And this is furrow length versus time. So our dashed lines are velocities. And we have a distinct slow, fast, and stalled region. So what we're going to try to do is use some of these sudden changes, the slow to fast, fast to stalled regions, and we're going to try to do this with a single coupled set of equations. And we're going to try to use this to understand what's happening with our cytoskeleton and membrane. So we're going to hypothesize that a viscoelastic model governs invagination. And so to begin, I've just outlined saying, okay, it's all about cytoskeleton, membrane, all this. And so what I'll do is I'll outline some of these materials that we're looking at and go into a little bit more detail. So fundamentally, biomaterials are really composites. And what I have boxed is the apical surface. Each of those bumps are microvilli. They are effectively stores of membrane, so this furrow can extend into the cell. And so our membrane is really going to separate our intracellular and extracellular spaces, whereas our cytoskeleton is going to provide the structure and induce shape change. And so on the left-hand side is an EM image. I put in a red circle there, one cell. And so all those little bumps are the little microvilli or the extra material that's being stored. If we look underneath of that through a cross-section very similar to the cartoon, we see a bunch of F-actin. F-actin is simply a type of cytoskeleton. And we see it building up in that area. And in fact, some work done by Figured has shown that F-actin depletion and microvilli and wrinkling tend to have a roughly linear correlation as these furrows invaginate. And so here's just a cartoon of a single cortex and single microvilli with this membrane and cortex. And for mathematical simplicity, we're just going to take all of those angled elements and make them horizontal and vertical. So as I kind of elaborated on already, we have a composite structure, cytoskeleton membrane. Applied to that is some external force, which takes this wrinkled membrane on top and straightens it out, makes it into a furrow. It's also moving through the cytoplasm. So we have some viscous fluid effects here. And there's some adaptation happening. So when we pull on this, there are different ways that the membrane and cortex can interact. First of all, the membrane is going to flatten out because you're extending its length. So we're going to see that flattening out. But the cortex can also move relative to it. So you can have, this is not a fixed point necessarily, you can have the cortex transmembrane protein slide through the actual membrane. So we're calling this our sliding cortex case. And then we have an unbinding cortex in which as you tension your cytoskeleton, your binding proteins, in this case, some form of active binding protein most likely, is allowed to unbind and you get more of a floppy network. Finally, our cell can adapt by assembling. And so when we look at that other image previously where we have all these microvilli, the cell needed to send up a bunch of vesicles to produce that membrane as well as to build that cytoskeleton. So we're going to include that in our model. And we can take all of these components and build a stress balance, a fairly simple stress balance, in fact. And we have our membrane and our cytoskeletal stresses. They're affected by adaptation. And we have some viscous fluid, or cytoplasm is our drag stress. And we have our motor proteins, which simply just pull on this thing. They put the input energy into the system. And so looking at our area here first, 1 second, we have our total area of a furrow is equal to whatever is wrinkled on top plus what is unwrinkled on the side. And our unwrinkled area is proportional to the length of the furrow. And then we have an unwrinkled apical on top. That's simply just the dome shape you see in the figure below and to the right. And so actually this figure is quite nice because this is an EM image showing the initial figure I showed you, where we have a cell with a bunch of microvilli, or a bunch of cells with microvilli. And as time progresses, that entire dome flattens out. It consumes all of its membrane. So from this, we're hypothesizing that area should be a limiting factor. And if you run out of membrane area, you will also stop furrow invagination. So we put this in there as well. And we say if you run it as effectively, you have the inverse exponential. So its wrinkled area decreases, our membrane stress increases until we get some equilibrium force between that and the motor proteins. And finally, we have a drag stress, which is a function of length and velocity, length of the furrow, I should say. And so now the more dynamic element here, I would say, is our reservoir stress. And that's really going to be our cytoskeletal forces. And shown in the top right, I have a base Berger model. This is the foundation of our model. We start with this and we cut things out as we find they do not contribute. And so the zigzag line under Kelvin Voigt, that's simply spring, consider it like a solid body. This bracket looking guy is a dashpot. That's just consider that like a fluid. And we're doing this in a lumped case. So that is to say, I am not looking at every single microvilli and tracking all of that. I am saying this whole body represents the cytoskeleton, and we modify these parameters to represent a transformed cytoskeleton. And we can do this in various ways. We chose to, by softening it, we can say we're adding, in a very physical sense, we're adding a F-actin in series with another F-actin, which is effectively softens. You get this pure softening mechanism. We can purely stiffen by just doing it parallel. And the modulus of the materials change as a function. You can see how they change as a function of each. So purely stiffening is linear, purely softening decays to 0. Finally, we couple our springs with our dashpots with an elastic viscous coupling. It's a very simple linear relationship. This is actually found from experimental measurements. And so we directly use that. So to begin, we start our investigation by not assuming any slow to fast mechanism. We just say, we have lots of good measurements from wet lab experiments. Let's use that. And we're also going to say, we're not going to consider what vesicles are doing to the system. Simply, let's just naively look at the cytoskeleton. So in this case, vesicles do not change with time. All of the membrane that it needs is there. And so in our shaded regions here, that's experimental data. And with the line, the colored lines, these are our various viscoelastic bodies or elements. You can see them boxed on the left-hand side. As a brief aside, you'll notice that one spring in the Maxwell is not boxed. We found very early on that has very little effect. We got that originally from some neutrophil data, which ended up not being relevant. So we have our three curves here, and we see our material properties drop with time. And so interestingly, one of our results is we compare everything to what we can measure in vivo. And so what we're measuring here is the amount of membrane area on the top surface. And so in the curves that we're showing with the gray curves, those are all fluorescent measurements of the membrane on top. And the blue line is our model. And so simply by using effectively a viscoelastic model, a fluid type viscoelastic model, and by directly inputting the material properties that we see for measurements, we get a very accurate representation of how the material on top changes with time. And when we look at our velocity versus time, we see a distinct slow phase, fast phase, and then finally a stalled phase. In the case of the stalled phase, that's actually due to our membrane runout. And we determine the amount of membrane that we initially need based on in vivo data. So just to summarize some of the initial findings, we find that viscoelasticity is highly likely to govern furrow behavior because we've been fairly naive with our approach so far, and been very data informed. And D'Angelo is actually the source of the data that we've used, where they measured at different points in time, and they fit it to this fluid type body, just measuring the applicable surface's material properties. Additionally, we do believe we have a plausible mechanism for the fast stalled phase, and that's due to a loss of membrane. So what we still need to know, what we originally set out to do, is determine what mechanisms control this viscoelastic shift. And so we have the two proposed mechanisms I described earlier. We have an unbinding cortex and a sliding cortex. And so what we do with the unbinding cortex is now we do allow vesicles to, in fact, exocytose. And so we let them go for the first 30 minutes based on data that we've acquired. And we allow them to have an effect on the membrane. And so an effect on the viscoelasticity of the membrane. So we allow them to add stiffeners proportionally. And then we allow the system to remove stiffeners as a function of tension. So if we remember, we pull on this thing, we can allow unbinding of our cortex. If we allow this unbinding, we now are softening it. So that has a linear relationship with time. And we get a reasonably slow region, a kind of torquey fast region where we would expect a little bit more linearity at some point. And then we see a stalled region. I will note that that stalled region stops a little bit early. That's due to some just numerical constraints that we put on the system. Next, we have a sliding cortex that we want to check. And in this case, vesicles are delivered at the exact same time. We had to get the results that we expected, we shifted when the viscoelastic softening happened. So in this case, vesicles softened the membrane. And we don't know yet why that is. And so we get this actually very pretty result here where we have a very distinct slow, fast, and stalled region. And so one of our objectives here is going to be now to go, well, we have these two plausible scenarios, each kind of have some weaknesses, some benefits. And our goal now is to use some loss of function experiments to better understand and better constrain our space to eliminate one or constrain where is a viable solution. And so returning to D'Angelo, they did a very neat experiment where they take a metal bead and they adhere it to the applicable surface. So you can see that in the top left. And then they put a magnetic force, or they put a magnet nearby and they pull on it. And so they see how the viscoelastic properties change with time. And so they do this in an oscillatory way. And so we're replicating that in silico. And so in the blue line here, or the blue box, really, those are a bunch of square waves. And at our baseline with the circles, that is our baseline stress that we use to make the previous models. And then that top black line right here, that's just a constant load that's equal to the magnitude of our, or the maximum of our oscillatory stress. And so with an unbinding cortex, we see that our viscoelastic properties change. That is significant because what D'Angelo found was that when they pull on it, there's no change in the slow or fast velocities or when they occur. If we're creating changes in our viscoelastic properties, that's going to change our velocities. And so this is no longer a very viable mechanism. And we use the same order of magnitude stress as they did in vivo. However, in a sliding case, also probably not terribly surprisingly, we saw no change at all. So at this point, we're going to go forward and get rid of our unbinding cortex, and we're going to investigate further the sliding cortex. Just to exemplify this in a little more clear way, I'm showing that there is a 70 to 80% loss in the, or full change, sorry, in the unbinding cortex case versus no change in the sliding cortex. So another loss of function experiment that we're replicating is a Berfeld-Binet experiment. Effectively, what that is the Golgi provides vesicles to the membrane, and what an experimentalist did, figured at all, they shut basically that delivery off at various times. So 0 minutes, 30 minutes, and 60 minutes. And they tested to see what would happen to the furrow. And so when we do this, we have the expected results for our 30 and 60 minutes. However, in our 0 minute case, we no longer get the fast phase velocity, and we should. This had very limited effect on, or had no effect on the slow and fast phase velocities. It did have an effect on how long the membrane grew. So this is distinctly a problem. We've lost our fast phase. So we dug around in the literature a bit more, and we discovered that the second compartment, the recycling endosome, sits above the nucleus. And that would allow us to then transfer our Golgi vesicles to the recycling endosome, and then that delivers to the membrane. And what we find is that we recover roughly our fast phase and slow phase velocities, and we stop at the right length. And so just for completeness, I return us to the original 4 plots that I've been showing a bit here. And we map out everything that's happening. And so we have some Golgi secretion for the first 30 minutes. We have some recycling endosome compartment accumulation of vesicles. The recycling endosome then exocytoses them to the actual cell or the membrane. And then we have our viscoelastic elements, which I'll remind you, we have a F-actin membrane correlation. And so we're keeping those correlated, and it's still shifted 30 minutes. We have some expected viscoelastic behavior, which is good. And interestingly, and kind of happily, our wrinkled area fits better the data, experimental data, and shown in gray, and our model in blue, where we have this initial decay for the first 15 minutes, and then a little bit of a spike up in the apical surface, and then once again a decay. And when we look at our velocity plots, or our length versus time plot, we have a slow phase, fast phase, and stalled phase. We also have this sort of slower decay region where we actually run out of membrane in our apical surface, and we're reliant on just how fast those vesicles can be delivered. It's no longer cytoskeleton controlling it. It is now the vesicle delivery rate. And so what we propose is on the left-hand side is what we were just looking at. And what we're hypothesizing potentially is that instead of just having the recycling endosome deliver all of its vesicles to the apical surface and build all these expensive microvilli that are really relatively stiff to pull apart, at some point, it starts delivering vesicles to the furrow directly. And there's good experimental evidence that shows where they label fluorescently the apical surface and they track where that color goes. And they say in the fast phase, this like unlabeled section of membrane just appears right here. And so we hypothesize that that is likely what is happening. So just to conclude everything, we suspect that a very plausible mechanism is that membrane stops the furrow at the end of invagination. Sliding not unbinding cortex accounts for the slow and fast shift, and the recycling endosome is a necessary secondary reservoir. Just very briefly, how can this be used in other applications? Well, we have T-tubules in our muscles, and that's used to direct a lot of signaling. And so if those do not form properly, we can get all sorts of muscular diseases. And so understanding how these large invaginations form can be applied there. From a morphological perspective, it's just interesting. A lot of species go divide 1 into 2, 2 into 4, etc. This is like a single cell to a tissue. And so finally, I would just like to thank my advisor, Dr. Rangamani, my co-mentor, Sam Ward, our experimental collaborators, Anna Sokak, and then the postdoc who helped me quite a bit working through some of these things, Maite Bonilla. Thank you. Questions? Yeah, I think so.
I mean, I think I know the project, but I have a more fundamental question. So how do you go from SpaceX to thinking about embryos of fruit flies? So what was that like for you? I realized I never asked, although I talk to us every day.
Yeah. So as an undergrad, I read some book on neuroplasticity, and that really kind of caught my interest. And it was very interesting to me how we can, it was like, I think it was like the brain that changes itself or something. This is the title. And so it was just like a very nice book. I started getting kind of into it, like put it off, got, job, whatever. And then I started working on composite structures, and I thought those were super cool. And it just kind of, I started thinking of like bones and being like, oh, they're not that dissimilar. I mean, they're dissimilar, don't get me wrong, but like if you want to like reduce it, you can mix a lot of parallels. And I was like, but they do a better job because they repair themselves. And I don't know. I'm also like deeply inspired by wanting to, make replacement organs. And the manufacturer in me just loves that, right? It's like, how do we take this cool stuff and mechanics and signaling and grow stuff, and then, help the world a little bit. So.
Let's talk later offline.
Perfect. Thank you.
Thank you again. Next, we have Dr. Jennifer Harris, who is a group leader of the Physical Chemistry and Applied Spectroscopy group at Los Alamos. She's as a trained respiratory toxicologist. Her research now focuses on developing non-animal models for studying radiation exposure, pathogen-host interaction, and toxicology of medical countermeasures. She has received 2 R&D 100 awards.
How do we pull this up from the drive?
Yes.
And one more question. Sorry, how is the what's the slideshow setting for it?
Okay.
Apologies. Oh, okay. There we go. All right.
Yes.
Awesome. Thanks so much for having me out. I got to meet a few of you this morning, and I was really excited about your research. Great. Thanks. I appreciate it. A little bit about Los Alamos. It's very, very unique. Most of you know it was founded for the purpose of developing and building an atomic weapon. So why on earth are there biologists there? After the war, the laboratory developed, tested, it grew new science areas. Our priority roles are still serving as a nuclear weapons lab, but there is a very, very strong biological capability there as well. A little bit about the national labs as a whole. Every lab has kind of its unique style, role, or niche. Up until 2011, we were, from the 40s all the way up to 2011, all of the national labs were designed to compete against each other. As you can imagine, that's not really great for science or even cost effective. But it kind of surprises me that it was up until that long that And we were here to do overlapping research. Today, we are not allowed to compete with universities. We are not allowed to compete with each other. And so really each lab has kind of its own role, and this is sort of a spectrum of different things. All the way from, you know, almost pure R&D science, all the way over to more of weapons and engineering. And then, you know, you have some of the special energy ones, like National Renewable Energy Laboratory, that really focus on alternate energy technology. So there's a lot of space if any of you are interested in the National Lab complex as a career goal. There's kind of a national lab for everyone. Our lab is biggest of all of the national labs. We have over 15,000 regular employees, and that does not even count contractors. We've got most of the budget, as you might imagine, is really in this weapons area here. Where biology falls generally is in the strategic partnership program, which is a very, very small budget. Somebody asked earlier about how many people work in biology at the national labs, and I don't know an exact number, but we are a very small part of it, probably 500 or less. Some interesting career paths. We've got, you know, traditional scientists, engineers. They do a lot of proposal writing, of course. We mentor students, postdocs. We actually have quite a few graduate students who work on site at the National Lab and then go to their home university for committee meetings and courses and things like that. Scientists, as time goes on, and this is true at the university as well, we have less and less time in the lab sometimes, and we regret that. Other folks choose more of a research technologist path at the lab, and that happens also with PhD level folks. They really want to stay in the lab. Good research technologists are worth their weight in gold. We have quite a few who are PhD research technologists. Research technician, we've got a lot of folks there too, but that's more of the high school or bachelor's level folks. And then we've got management. I took a turn down the dark path towards management in the last year and a half, and it's actually kind of fun. So these are, but it doesn't mean that we can't wear some of these different hats at different parts in our lives. Every national lab has challenges and opportunities. Some of the ones at Los Alamos, I mean, we're really a stockpile stewardship lab. We make nuclear weapon kits. It's our primary mission. So sometimes folks like us in bioengineering and biotechnology, we have to take a back seat to that. Many stable funding sources require a security clearance or citizenship. It's not impossible to get there, but it makes it harder. We have lots of bureaucracy and aging facilities. Every time I come to a university like this one, I think how lucky you all are to work in a beautiful facility like this with the latest and greatest of technologies. And sometimes the funding is really tightly tied to the laboratory agenda. Like I mentioned, each lab has to have its own little niche. And so we have to sort of stay in our lane, which is, you know, it makes for some interesting challenges scientifically. It's how do I do what I really love to do and fit that niche? Some great upsides, you know, wonderful, wonderful, amazing coworkers. We can really reinvent ourselves. Sometimes when there's a need, you think, well, I'd really like to try that. The person who spoke before me who went from one field to a completely different one. And that, you know, I think there's a lot of opportunity in the national labs to do that as well. And, you know, Los Alamos, it's a fun place to live. It's very small, and we don't have traffic like we have here. But a lot of people don't like it as well because of that same small town feel. How did I get here? Why do I work there? I really am fascinated with understanding radiological phenomena, defending against chemical, biological, radiological threats. I mean, that's a passion. Make sure that when something happens or before something happens, we ideally have a plan and preparation for it. We have a dark past with legacy sites. We want to help clean those up. There's a very big role in the biological sciences for that. And also really informing public policy. I think kind of that's maybe what we all want to do on one or another scale. I've not been a very direct scientist. I didn't go right into undergraduate and go, wow, this is what I want to do with my life. It took a while to get there. I don't know, how many of you had a career before you went to grad school, or did most of you go straight through? There's a few of you in here. I dropped out of college for a little bit, ran off to Europe, did some archaeological excavations, and then ended up working for a museum that sent me around traveling around Germany. At some point, I realized that wasn't going to be a forever career. And I thought, I really need to do something where I'm going to change things, help people. So I went back to school and studied respiratory toxicology. And I ended up after graduate school at Los Alamos, where I worked with viruses, started in on engineering with robotic systems, became a board certified toxicologist, and started working in the bioengineering field in some of the early days of organs on a chip. We've got a long history of human research. These are plastic Nor phantoms. Phantoms are used for radiation research, both computational phantoms and actual real physical phantoms. In these phantoms, different dosimeter devices were embedded, and we were able to determine a little bit more about how radiation moves through the human body. It informed quite a bit of medical research down the road, but also weapons research as well. Some of the microphysiological systems that we've developed are lung, heart, liver, neuromuscular junction, skin, and really all of these have a national security driver. Now that we are in a more modern era where every university has different microphysiological systems that they're developing, we're doing less and less the development because that's not what's needed. What's really needed is to utilize that national security driver and figure out, okay, so what do we do with this? How do we help solve national security challenges? If I can get that off the top. I'm just trying to see if I can make this disappear. Oh, there we go. Hide video panel. There we go. That's better. Now you can actually read the titles. I'm sure those of you who are some of you are in bioengineering, some of you are in different fields. We are all aware of these changes to toxicology and the drive to reduce animal use. APA, quite aggressive starting this year, or no, this coming year, with their changes towards non-animal models. And the European Parliament has already started moving in that direction. They have very large organizations in every country in Europe now trying to understand how do we replace this test, and another test to make sure that we can continue to work with creating new pharmaceuticals and using non-animal models to test them. We've also recently had the FDA Modernization Act, which is interesting because it allows for preclinical testing in human relevant models, but it does not require that. I mean, I have this question of, it's sort of this paradigm of, well, animal studies are used for preclinical testing and human safety data because we understand. I think the first written records that we know of in toxicology in animal testing go back to, I think it's 500 BC. So we have 2,500 years to catch up. And we have to use every possible tool we can to really make this work and understand what is this organ's best role, or what is this computational tool's best role. But before we put the proverbial lab cart before the horse, we really need to demonstrate that tissues and organs are fit for their intended purpose. Let's say, for example, you want to work with the FDA and have an organ validated to replace a specific test in preclinical testing. You need to demonstrate that, and they do have an alternate methods working group, which is very, very helpful. But one of the things that they do tend to see coming a lot from our ilk, meaning the bioengineers or biologists, is that I've got this great tool and it's going to do everything. And they immediately think, well, that's disappointing because there are 18 different tests, for example, for reproductive toxicology. Each of our systems or things that we build may be able to replace one at a time. So we need to think about that and keep that in mind. And I think there is a little bit of humbleness that we need as a community to help really bridge that gap and work together with folks like the FDA. And we also need to use our computational omics tools as well to demonstrate, hey, this tissue does that. You know, for example, computational tools where you have to account for missing organs. If you have, this is an example of a lung organ on a chip, and you know, you've got many, many missing organs there, and how do you recreate what that might look like in rats, or what that might look like in humans? Computational tools can help go a long way there. So in my lab, we try to use our human surrogates or computational tools as crash test dummies, and that's our goal. But of course, you know, we have to first figure out what are they good for? How do they work? How do they compare? A couple of multidisciplinary projects I'll share with you today. One is called HEROES, Human Exposure Radiation Use of Organ Systems. And another is part of our Department of Energy response to COVID-19 pandemic, which was called the National Virtual Biotechnology Laboratory. Some thoughts. Haven't we understood everything about radiation? There's been lots and lots and lots of animal studies, but still, no one agrees. If you look at the curve on the right-hand side here, curve A is showing, you know, we've got a response that actually seems to be progressive. And then all the way to curve D, which is called hormesis, radiation hormesis, which is a phenomenon where we think that there may be actually at very, very low levels of radiation. Think about it, we're getting very, very low levels of radiation right now. May actually be beneficial and required for certain functions. And so, no, you will not find a set of toxicologists or radiation biologists who agree on which one of these curves is real. So for one of the projects that I led, we integrated 3 disciplines, chemistry, modeling, and biosciences, to identify what sorts of radiation doses should we be looking at, what types of, how accurate can we get on our doses, and also how do these organs stack up against, say, human responses? We've addressed some of the questions of how do we calibrate exposure at low levels, and also for very, very tiny organs. You know, what's the absorbed dose by the biological materials? The absorbed dose is going to be much different that if you've got something that is a tissue versus a piece of wood, for example. And the goal really is kind of, how do we rapidly triage people? Is that going to be even possible? We've got some interesting multidisciplinary teams that make this happen. We have cesium sources and rooms that are available for gamma irradiation. Some examples of things that we did would take, we culture our organs for specific tissues, and we'd drive them over to these gamma rooms and irradiate. We'd have some that drove along and didn't get irradiated, and some that stayed in our lab. It's a big campus, so just like here. So anecdotally, it's also interesting to see just driving these tissues around does change their behavior as well. But we followed up with transcriptomic analysis and traditional analyses as well. Some things that our computational team were really helpful with are things like irradiation of samples. I mean, there are guidelines from the International Council on Radiation Protection on what doses might be, how to calculate doses, but there wasn't anything for something this small. And so we used Monte Carlo and particle computation to optimize our geometries for radiation exposure. And even with the best computational methods we had available, if you stack things up or put things behind or next to, even in a very small area, you can get variation in the dose that a sound is going to receive. And so we use this computational method to try and figure out, okay, well, we can stack this up, but let's exclude this sample that's in this position in our gamma beam, because that's clearly not getting a similar dose. Human phantoms are where we started from. So you saw the pictures of the plastic man, and there's also computational versions of these. For medical research, they go down to the organ level, and for this project, we took it down to a sort of a 1 centimeter by 1 centimeter tissue level. Some other questions that we needed to address: how do we generate real world materials? And by real world materials, I mean ones that I hope you actually do not ever breathe in the real world. Fallout simulants, things like uranium and other plutonium, other metals that can be in an event like a dirty bomb. We wanted to find out what's happening at the biological interface. How do you get these metals to dissolve in biological media? That was a fun one. And then, of course, what's the biological outcome particulate exposure for things like fallout. There was a question earlier about what makes a nanoparticle toxic versus another one. Often it's shape, roughness, size, length. Those are all different factors, not just the material. And so when we created different particulates, controls, and others, we found that they used ball mill to machine them. We found that they actually came out looking quite different in shape and size. And so those are things that, you know, we still need to tease out, is how does, and these are not nanoparticles, these are microparticles, but they do tend to behave in some similar ways as nanoparticles. How do we determine things like aerodynamic particle sizing in radiological samples? Our aerodynamic particle sizer wasn't in a radiological lab, so we had to run controls through and use computational methods to identify what is the likely aerodynamic particle size. Our dynamic light scattering instrument was in a radiological lab, so we were able to run that and get measurements for both diameter with SEM and also diameter with DLS. So that helped. Our goal really, you know, if you've got something that's going to land in your lung, that's about a 1 to 5 microns in diameters is what's likely to land in your lung of anything, like pollen or things that might be flowing around today. And we also looked at isotopic content. So, you know, how does the isotopic content affect our tissues? One thing that we did early on is to even just look at cell type. What cell type we put into our organ systems really, really matters. We found that the IC50 or the even the effective concentrations of different types of uranium, be they depleted uranium, which doesn't have a lot of alpha emission, all the way up to highly enriched uranium, it actually changed quite a bit depending on what cell type you put in there. Of course, we all, you know, it's very nice to work with cancer cells because they grow and they, you know, you have a hard time killing them. But interestingly, depleted uranium did a really good job of killing A549, which is a lung cancer cell line, which we thought was unique. But after we went through many iterations of these experiments, we realized that what was happening, and different follow-on studies, we realized what was happening was metal toxicity, not so much the radiological toxicity. We performed transcriptomic analyses on our samples to identify relevant genes and compared that with what's known in medical literature for our doses. And we also confirmed our results using, you know, traditional methods. H2AX phosphorylation, for example, is a very classic sign of radiation exposure. And we wanted to see how low of a dose could we use on our systems and still see these responses. Some highlights of this study is that we used a lot of tools built for weapons effects, and we actually were able to optimize our doses for small tissues. I think that's a really big win. And also comparing with those known medical effects, it helps us ensure that the organs are fit for that role. And not all of our organs ended up being appropriate for that job. So it's okay to say that, you know, hey, this lung tissue was working out, but only if we use these types of cells in it. I'll share some other stories too about sort of, I guess we all have a what did we do over COVID break story. we had a choice, and I think you all probably had a choice, is that you could do something that was considered essential, or you could sit home and do something in front of the computer, which also might have been essential too. But I know that I wanted to come into the lab and I wanted to come in and work. And so I volunteered for one of these projects, and my boss said, you're going to work with these guys from engineering, and they do explosions. They do CFD, computational fluid dynamics for explosions. And then there's a couple of guys over here, they do some aircraft engineering. And okay, well, and then they said, well, there's this ventilator company and they want to know if their ventilator is doing the right thing for COVID. They had a passive ventilation. So if anybody knows someone who has cystic fibrosis, you whack the kids on the back to get the mucus out. There's also these chest wall oscillators shown here that sort of thump the lungs or thump the chest wall to help loosen mucus. And because COVID-19 had so much hyaline protein and mucus, we wanted to take a look at that issue and see, well, is their ventilator doing much? My colleagues were a little surprised when I came into the lab one day with some sheep lungs and said, well, we're gonna hook this up and see how deep with some dye and see how deep this goes. And it didn't go very far. The lung is described in generations, so each branch is another generation. Even with percussive ventilation, we only got the dye maybe three generations deep. And so how do we change that? Can we change that with frequencies, different pressures? And of course, we don't want to cause ventilator injury. That's not good. No matter what, if you go on a ventilator in this world, you only have a 50% chance of ever going off. That's bad odds. So we wanted to help this company improve things. It got even weirder. The president of the company ended up hospitalized with COVID and on a ventilator during our project. So we weren't able to work with him directly. We had to work with some of the people that were part of this team. And so we wanted something that was long-term for ventilated patients. We set up kind of this end-to-end lung system analysis where some of the aircraft engineers were doing CFD on a model of the lung that had a tracheal tube down it. So we found that nobody had actually done a model with trach tube. You know, you think about airflow in and out, but there's this little bead here that is preventing air from going out. So how does this look, and where's the turbulence? And there wasn't really any good mesh for anything past maybe the 7th or 8th generation. So we had to create that as well. We also had an acoustics team that I got to work with that was able to look at different hertz of percussion. And we also did some laboratory organ work where we had our team help us compute particular stretch for ventilator injury. And so here's an example of simulating vibration frequency and mucus clearance. I tried to culture mucus because if you're culturing lungs, you're actually, that's a kind of a byproduct that you throw away. But it's a very small amounts. And so I said, well, here I've got this little bit of mucus for you. Can you study it? And they said, no, that's too small. So what I did is went back to the literature and found and talked to some of the scientists at the lab who were rheologists. Well, what can I use that's going to be like, or have the rheology of, or they were tribologists, I guess is the proper word. What can I use? And we found this compound that makes hair gel. The hair gel is made out of cortical-membrane. And we measured it to see, does it have similar properties? We were able to use nice large amounts in our 3D models of the airways to test our progressive ventilation. For the ventilator injury, this is where the aircraft engineers came in, and when we think that we're getting turbulence in different places, they went even further. You know, so if when you normally breathe in and out, you are going to have some turbulence in the lung, but putting the trach tube in means you're going to have turbulence in different regions of the lung. And some of the things that they found is that also thinking about changes in compliance are going to be important, and we need to be able to measure that on ventilator patients to be able to, and you can see that to some extent with a medical exam. Learn a little bit about that, about the compliance of patient's lung. But they also discovered, you know, that's really going to be important as far as stretch goes and what happens to prevent ventilator injury. So we also used our microphysiological systems based on some of the stretches that they said they would be looking at in the deep lung, and to try and mimic our physiologic stretch. And you know, we were able to take that and also use a percussive method as well to see how is that going to change our cellular system. One thing that we did find is that, and it makes sense, is that some of the frequencies that we tried were absolutely unsuitable for keeping our tight junctions together. And so those are also, you know, there were two frequencies that were better for mucus clearance, but one of them was absolutely unsuitable for systems of cellular life. So that better tested on our systems than in people. Some highlights there. We were able to help the company with recommendations so that they can make a better ventilator. You know, it's sadly, of course, too late for many of our. We probably all know someone might have lost during COVID. It's a bit late for that, but I am glad that we're able to help someone in the future. And I think the computational methods and the people I got to work with, they really helped me ensure that I was able to at least run the microphysiological systems a little bit more analogous to what's actually happening. I'll close with some soft skills for multidisciplinary research. It's just sort of some useful things for all of us. Be willing to teach your colleagues and let them teach you. I had so many folks in different projects who asked me what a biomarker was, or you know, what are the different levels? Where are you working? Where is your space? And you know, often with our microphysiological systems, we're kind of working in this space because we can't recreate every single piece of an organ. Sometimes we can work on the whole organ level if we have a human sample, but for the most part, you know, most of us are working in this realm in bioengineering, and I think that it helps your colleagues in other disciplines to really understand that. some fun things is, you know, be careful of height. I think there was a time and everybody said, oh, well, you know, tissue chips, they're going to do everything. And then we kind of got this sort of trough of disillusionment from regulatory bodies like the FDA, who said, well, hold up. We have rules here. And we also have specific tests that you need to replace. And I think we're getting towards this plateau of productivity here, where we really are starting to see what are some of the benefits, and we're starting to get International Council on Harmonization interest in replacing some of our experiments. Sometimes, you know, when I see this kind of Gantner hype cycle, I think, I mean, every technology has this. You know, I wonder where we are with AI. You know, there's a lot that we can use artificial intelligence for, but there's some things we have to be careful of, especially the lack of negative results that are going into some of our models. And also remembering to acknowledge those who helped you. These are some of my amazing students and postdocs and people who've worked with me. This work didn't appear in a vacuum. There was a lot of people helping on this. And also, don't forget to build relationships with your sponsors. It's interesting to think of our sponsors as this sort of like, they're over there. They're in DC or they're somewhere else. And yes, it's true that the lifelong R01 is probably gone. Those days may be gone for many of us, but it doesn't mean that there's not a lot of other sponsoring organizations and program managers wanting to fund you and build a relationship with you and keep funding you. So those are good things to keep thinking of. Any questions?
So from your first project, how are those results being used when you were looking at how the different types of radiation affects different parts, the different organs? How are those results used today? Like in what parts of the world, things like that?
So from the organ work at this time, we're still just doing very, very basic research. We're moving into a realm where we are looking at excretion. And this is actually in a skin model. We're looking at excretion of uranium and plutonium. So right now, if you are a worker in a plutonium facility, you do what's called bioassay, which means your urine sample is analyzed and they cook it down. It takes like several weeks, and they perform mass spec on it to see if you have inhaled any plutonium or uranium. They do that for uranium workers as well. Right now, what we're trying to do is recreate some testing abilities where we could do a swipe of the skin. So we're looking to see if uranium and plutonium are excreted in the skin over time. So we are using them, but in a little bit of a different path, and it's more applicable to the lab or mission, if you will. Yes.
Perhaps startups emerge or split off from your groups, like if you, for instance, you know, like develop like some kind of like more effective mask, you know, these kind of particulates, like would you be able to, like, or I guess like you got a national lab, does that have any kind of like prevention from, I guess, going mainstream?
There are startups that do emerge from national labs. In this realm, at least for the mask situation that you described, we were actually helping a different project, different set of people. But there was a set of people helping look at a company that, or help a company that was designing sterilization techniques for masks so that they could be reused. And that was driven solely from the fact that we just didn't have personal protective equipment at the early days of the pandemic. So a lot of, I think people can take it a different way. There are some people who do leave the national lab and create startups. It's a little less common, I think, at the national lab than it is in, say, university situations. We don't have quite the incubators like a lot of universities do.
I'm from New Mexico, by the way, so yeah, nice to glad to meet someone from Los Alamos.
Oh, nice, nice. How do you what do you think weather differences? Which is better weather, San Diego or New Mexico?
If you like snow, then New Mexico. You know, we don't get snow out here.
Yeah. Yes.
For the CFD models for the lungs, were you basing it off like imaging data, or was it like a branching scheme?
So what we did is we bought the first 7 generations of the lung as a computational model, and then we added generations to that based on there is some empirical data out there, but it was kind of a painstaking thing for the computational modelers to stitch these grids together. Yes.
That with a lot of the knowledge that we've gained over the past 50, 60 years on radiation, that we're in a better position now than we were 20, 30 years ago with respect to other nations continually testing out in certain areas. This radiation is cumulative, and in these environments that are getting tested, like kind of like where are we at in this story of our exploration of these powerful sources?
I still think the best solution is prevention. I don't know if anybody read Annie Jacobson's Nuclear War book that just came out this year. It might be an obscure audience. It's interesting, and then you get to the last chapter and everything is spoiler alert. It doesn't go well. And I think, you know, it's good to have books like that out there to kind of scare us into, this is the worst case scenario that could ever happen, and we do not want that to happen. As far as tools that exist to help if something did happen, there's a really great tool called Henry from the Department of Defense that looks at prompt radiation and how can you help the most people in an area that's been affected. In the HEROES project, and I didn't touch on that, but we also developed a computational tool for fallout modeling and inhalation exposure as well. It's cumulative and it isn't. It's because most of our DNA damage, I mean, we have, I think it's 50,000 radicals per cell per day. Maybe it's per hour that each of our cells is making. So, I mean, we have DNA damage that is occurring all the time. However, and most of our systems are overcoming that. However, yes, once a cell divides with a damaged nucleotide, that is cumulative. So these are things that, you know, we do want to keep in mind, but not just from a radiation standpoint, from a whole life standpoint as well. You reach a certain threshold where it's no return. So I'm sorry I don't have a better answer for, you know, what other countries are doing. Because we know there's, in the newspaper, there's things out there that are frankly scary. And I think the best prevention is diplomacy. Sorry, that was dark. I'm a toxicologist, so you know we talk about poisonings all the time. That's just, you know, sorry. Yes.
Yeah, I know you mentioned that the biological research, such a small portion of the research going on at Los Alamos, is kind of driven by national security needs. Are there any other national security needs at Los Alamos that have this biological research being done besides like radiation, or I guess COVID-19?
Sure. There is energy research. There's also countermeasure research. So if here I am going dark again, but for things like the risk of a terrorist attack, chemical weapons, we do a lot of research on what pharmaceuticals could be good countermeasures to different types of chemical weapons. And there's also some basic research, to be fair. You can't get to these points without doing some basic research to really understand some of the phenomena that we see. There's so much more. We have a huge genomics capability, immunology capability. We've got environmental sciences as well.
And climate change and ocean modeling as well.
Thank you.
Thank you.
That we thank Dr. Harris. We have a certificate for you. Thank you so much for coming to talk with us.
Thanks. I appreciate it. I appreciate you inviting me.
It's our pleasure.
Oh, sorry.
We will now take a break for lunch. Lunch is in the other room here. There are some posters up. The trainees will also be having lunch now. We'll also have them talk about their posters later. But yeah, we will resume at 1 o'clock as of right now for the academic panel.
So I don't see my own name.
I feel like I'm doing it. But I think I'm not.
It's so funny that I
My eyes are
I know.
I can see.
Oh, you guys have
They are.
Well, it's more. It's not. I mean, like, it's not even. I'm like, yeah, I'm tired. I'm tired. I had a meeting yesterday. I was like, I need to get back. So, I'm like, it's gonna run. I'm not gonna sleep until it runs. No, they ran. Thank God. Yeah, I don't get it. Oh yeah, are you kidding? I have to.